All right, so it is my great honor to present our next speaker, Dr. Robert Gentleman. Uh, those of you, it's hard to give an introduction to Dr. Robert Gentleman, but I'll go ahead and take a try at it. So he's executive director of Harvard Medical School for Center for Computational Biomedicine. He's also one of the creators of the R programming language, a founder of the Bioconductor project, and he served as vice president of 23andMe, where he helped launch their therapeutic division, and was the senior director for bioinformatics and computational biology at Genentech. He's head of computational biology at Fred Hutchinson Cancer Research Center and held academic positions at Harvard University, University of Auckland, and University of Waterloo. Dr. Gentleman's research interests are related to genomics, machine learning, data visualization, and the application of statistical and computational methods to study human disease. It's our Pharma's honor to welcome Robert Gentleman. Thank you.
Did a great job of it. And I really want to thank the organizers for inviting me to talk, and all of the audience for coming to listen to the talk. And I enjoyed Dustin's introduction quite a bit, except for the last slide. I'm pretty apolitical, and in case Dustin didn't know, I'm Canadian, so it's unlikely to actually ever be achieved, unless U.S. law changes, of course. And then there's much better people. Yeah, so my career has spanned a few decades now. I started off in academia, then I transitioned about 14 years ago to industry, spent half a dozen years at Genentech, 5 or so years at 23andMe, and now I've come back to academia because I think there's still some interesting things to do. I enjoyed the work that I did with Rossi Haka at the start of R, and working with the folks that are still our core and developing the central part of it. Similarly, the folks that I've worked with at Bioconductor and who have sort of taken it on to new heights. And I've sort of came to a conclusion a few years ago that we're not doing as good a job on data as we could. And so when the opportunity came to return to academia, and especially to Harvard, I took that opportunity, and in a sense where R was sort of a pure programming language play, Bioconductor was how do we solve problems within one discipline sort of play. This current work for me is really going to be focused on data and how do we use data. And I think that's the thing that's really changed in the world in the last little while. And there are some opportunities that might not be as obvious, and where we as a community, and I think that's part of what I really liked about Dustin's intro. We are a big community, and that's sort of been an amazing thing to have happened. And I do sort of really appreciate and want to thank the people who go to all the effort to make this such a good community, both within R, within Bioconductor. There are lots of groups doing all kinds of things that just make this a better place for us to do science. And those people really do deserve some substantial credit for that effort. Some disclosures before I get going too far. I do consult for and have options or shares in a number of companies, including 23andMe and HubMap, Maya Health, and Clover Therapeutics. There are a couple more that are still in stealth mode, so I don't really want to publicly name them. Now I have to switch here. All right, so outline. So I'm going to talk a little bit about the challenges, the way that I see things. I'm going to start, the organizers asked me to spend a little time on the history of R and what we did, and I'll definitely start there. And then I want to talk about the two of the things that we're working on a lot here at the CCB at Harvard, which are sort of phenotype problems, database problems on the phenotype side, and then data problems and challenges on the omics side. And then I'll whine a little bit about the inadequacy of current funding models, whether they're grants funded from any organization or philanthropic funding, they all suffer from a couple of defects that make it very hard to do large-scale shared infrastructure development. And then before I start, just two shameless plugs. So one is we are hiring at CCB. I put the website on the first slide, and it's pretty easy to find it. And so if you are looking for work, have a look and see what we have open right now. And then we're also discussing at both the R Foundation level and the Bioconductor Foundation level how to raise funds. So if you're somebody whose organization is large and is thinking about funding some of these things, I'd be happy to hear from you. All right, so software. So data-driven science, that's the world that we're really moving to, and I'm not going to try to make an argument for that. I think most people on the call are there, and we know that there's a whole bunch of stuff going on where data has just gotten bigger and better, whether it's from, epidemiological side, from government, from population, from humans, down into biology, neurobiology, you name it. We are collecting more data, it's more complex, and we're using it and models more than we have in the past to try to learn things. And so what do we need? Well, we need something that I would just call data technologies. I'm not sure there's an accepted name, but we need some way to store, organize, and then provide access to these very large, complex data sets. 20 years ago, in the early days of companies like Google and others in Silicon Valley, there was this idea that you just sort of could take all this data, dump it somewhere without much curation, and that somehow machine learning would figure out what was going on. And I think we've in the interim realized that machine learning works really well on well-curated and established data sets. It's not working that well on unorganized, uncurated data. And so I think most of us prefer to have our data well curated. I'm certainly in that group. So we need ways to do that. And the scale of the data is so large now that you can't really do it easily on your laptop. And even for some universities and companies, the infrastructure needed might be quite substantial. You need analytic tools, and that I think of things like R and Python, programming languages, and other things that help you basically transform that data through some kind of filtering, modeling phases to get back stuff that you want. The data are large and complex. As I said, we need visualization and tools that will let us do that. And then we prefer to have some kind of interactivity so that we can do something and see what changes. And those are sort of the building blocks that we need. And we have lots of tools that fit into those. It's the putting them together that's sometimes hard. R, well, I think R is pretty well suited to the second one of those. I think it's a good tool for data science, statistics, computational biology. Essentially, if you have data, you want to apply discrete actions to that and transform and extract information. And you want that information to help you sort of, and I say specific scientific question, it could be any sort of question, it could be a business decision, et cetera, but you sort of want to process that. Discrete actions, so we want to do different things. That really suggests packages that are relatively self-contained, or at least to me it says. Large complex data suggests the need for models, often using matrix algebra, complex machine learning toolkits, et cetera. And the transform and extract is scientific program. Is basically scientific programming. Scientific programming is really not like any other kind of computer programming, and I kind of learned that in my sort of spin through Silicon Valley, that there are really fantastic software engineers there, but most of them have no idea how to actually program a scientific algorithm. And so these two don't overlap that much. And that in part, I think, is why R has never really been something that people who are trained in classical software engineering show much interest in, because it's not really doing what they want to sort of think about or how they think about the world. It's really pretty different. Past R, so a small diversion just to give you a sense of where we started and what we were doing. In 1992, Ross and I met in Auckland, New Zealand. I was there on a sabbatical leave, and Ross had returned from having studied in the United States and had I think one or two academic positions, and then came back to take a full-time position in Auckland. About a year later, I decided that I would take a position also in Auckland and moved there for about just about 10 years. Just to give you a sense of why some of the things in R are the way they are is because the world, as you see it for computer science, wasn't really the world that we had then, right? When I first got there, there was really no internet that you could rely on. You couldn't transfer files from North America to New Zealand in any reliable way. You couldn't even get email reliably, depending on which of the networks you happened to be on at the time. Java didn't exist. It was a language called Oak at the time. Linux hadn't been created yet. And so we sort of ended up with laptops that either ran a Mac operating system or a Windows operating system, and there was just about nothing in between. And so now we have all kinds of opportunities. Relational databases were barely around, and when they were around, they were really thought of as things that ran industry rather than ran science, because the data in science was not very big. It was on the order of thousands of observations and hundreds of features. And we tried to design a language in that world. We like to use a sort of scheme-based interpreter for that. And the scheme-based interpreter we'd written in C. So we were sort of, the language we implemented in was C. We used that to create something that looked like a language called Scheme. And then we put a syntax on top of Scheme that looked like the one that you're familiar with in R. And so that sort of three-level thing actually turned out to be a reasonably good idea, and it helped us abstract out ideas and implementations. And that kind of layering and abstraction is important for all kinds of things. We used a version control system pretty early on so that we could both work on it, make changes, and check on things. We started with something called CVS, which has no relationship to the drugstore here in the United States. And then we switched over to Subversion, and that to this date still does everything that we want to do. Git is, kind of interesting, but the level of complexity that it allows is not something that we've ever found necessary. And so there just hasn't been a switch from Subversion over at least up to this point in time for R. Bioconductor has gone to GitHub and as their main repository. And this sort of source code control, I think, is really essential for any kind of paper or shared development. And we then formed this group called R Core that got interested in the work we were doing. We put it out as open source, so the Free Software Foundation had existed for some number of years ahead of time, and we basically applied or said, hey, we're doing this, and Richard Stallman said, yeah, great, you can be in the club and be one of our GNU projects. And so we had a license put on that was consistent with that. There was a language called S around, and once we started to do stuff, we were forced into being compatible with S for most things, and R and S are essentially compatible at this point in time. Nowadays, it's more S that gets driven, but it does still exist. We used a very different mechanism for packages than they did, and that I think they've now adopted ours. We developed documentation in the package framework that allowed us to do QA and QC, and that was an essential part. And then vignettes were really something that we developed in Bioconductor, and the idea of vignettes was really to separate out this sort of a key concept that I think is sometimes missed. So when I write a package, it has a whole bunch of functions in it, and those functions do something. So I should have a manual page for each one of them that tells you what the inputs are and what the outputs are. And that's great. But it doesn't quite get you far enough, because anybody that sees that won't necessarily know how to put all the functions in a package together in a way that produces an output. And so vignettes are another style of documentation that goes with a package. And the goal of them is to say, here, if you're a user of this package, now you can see what the functions do. Here's how I think about you using it to actually achieve something. How do you put those functions together, potentially with stuff in other packages? And that's turned out to be also a super valuable tool. It expanded to being, whole books are written in that, and our studio, and I keep forgetting what their current name is, developed some of these, or people developed things like R Markdown and Bookdown, and suddenly we're in this world of almost reproducible research. We're not quite there yet because we haven't solved the data problem, but we'll get there. Just want to make sure people understand that R is far from static. Anybody that used it in 1999 or 2000 to analyze microarray data knows that the speed at which it's operating now is orders of magnitude faster. We have a very complex set of object-oriented programming tools and getting more. We're moving into the large vector world pretty quickly. Thomas Calabara has very recently done a huge amount of work to get us to be UTF-8 based, and we'll see that sort of benefiting people, especially for internationalization. Luke Tierney, among many other things, put in the bytecode compiler quite a few years ago, and bytecode compiling gets us a fair amount. And we can work on the bytecode compiler if we want, and that sort of benefits everything. Image analysis and processing, there's some stuff in there. We're starting to work more on that in Bioconductor, and in most cases, we're trying to interact outside of the R language with tools that exist and are useful. And then packages give users lots of ways to contribute. That turned out to be something we didn't really think of in the first place, but it's the thing that lets people sort of move from being users of this to developers to sharing their ideas. And it's been remarkably successful with thousands of packages around. Where do we want to go? So our biggest problem, I think, is to make sure we get some kind of sustainability, as the average age of our core seems to go up by almost a year every year. And so we are recruiting some younger people, but we do need funding and other help in that place. There are lots of fields that are evolving and lots of different algorithms that are advancing, and we need to find ways to work with people to keep up to date. Okay, so that's sort of the R history one, and probably took too much time, but I did want to sort of spend a little time on that and give you a flavor for how things happened. Where do we need to go from here? Well, we really need our papers to be much more computable. It shouldn't be the case right now that I find a paper in Science or Nature or in an epidemiology journal based on a well-known public data resource, and that I can't actually reproduce the results in that paper without a huge amount of work. I should be able to find the code that they used. If they addressed a particular public data resource, I really ought to be able to get that code, the same data resource, and reproduce the paper. And sadly, that just is not the case. One of the things that we're trying to do at the CCB is to show some examples where it is the case, and then hopefully that will help others start to see that it's not as hard a lift as it might seem to be at the start. And there's lots of stuff to be developed in between, and I'll show you some of that. Too much energy into resetting up stuff. There are databases around at times, but really just not enough, and it's very complicated. And we need to have some more community building. So there is a fair amount now, but we do need some consensus solutions on some of these things. And so meetings like this one, where when they're in person, let us chat more than we have been able to for the last few years on some of these ideas. And so hopefully we'll find ways to form groups around some of the thorny problems and try to move forward. Data challenges. Okay, so here I'll just go into some of the ones that I know about and that occupy my attention. So large complex data is now available. If you remember back a few slides, I said a big data set had a few thousand cases and maybe 10 or 100 phenotypes or columns in that data frame. And now we're into databases that have millions of individuals and tens of thousands of phenotypes. And so when I was at 23andMe, our data tables were 10 million people by tens of thousands of phenotypes. And so you could really needed some tools to help you reduce that data down to a manageable size. Biobanks, so resources that give me information about humans, the UK Biobank, FinGen, All of Us, NHANES. Insurance data sets, it's possible to buy large insurance data sets so you can get most of the, or much of the insurance payment information in the US, which is pretty informative about health. And then other countries like Denmark and more of the Nordic countries have large basic public health data sets. Hospital records are becoming important in epidemiology and computational biomedicine. We do GWAS studies at scales that were never thought of before, multi-omics, and we'll go into that in a little bit. We have lots of social media, smartwatches, health devices, in-home monitoring, and companies are working on all kinds of ways to help improve our lives using these things. So that's where we're at. This data is gigantic. And then it doesn't take very long to realize that, if you're an epidemiologist and you study something, you find it in the UK Biobank, and then you'd like to replicate it in FinGen or All of Us or NHANES. And how do you do that? How do we think about doing meta-analyses? And one of the problems that comes up pretty quick is what I think of as the phenotype problem. So we don't have standard phenotypes. Type 2 diabetes doesn't mean the same thing in every database that you look at. And we don't know whether they measured things with equivalent units, were the populations that they studied similar. And every time you go into some of these data resources, you're all on your own. You have to solve every problem, from the first of how do I download the data and put it into a database, up to how do I interpret the data. And that's just not sustainable. We can't succeed if that's what we're going to do. So other phenotype problems. So in pharma, for the pharma folks, one of our problems, I think, is we don't have enough of them. We study complex diseases like type 2 diabetes as if it's one thing, but there are many different types of type 2 diabetes. And even if you don't think that there are many different types of that, there are very different disease trajectories that people follow. People who have a elevated hemoglobin A1C can change their diet and exercise, and suddenly they'll revert to looking like they're almost normal. But there are other people who have a condition called LADA, where essentially they're going to end up looking like a type 1 diabetic. Their immune system is going to reject their beta cells and wipe them out, and they're going to be insulin dependent, and no evidence that exercise or diet has any impact on that trajectory. And so we need to understand those. So we need, I think, in pharma really to think about drug development as we go forward, more in terms of disease trajectories or a long-term path of what's happening and what separates people. And almost all human diseases are complex in that nature. And then we don't have enough exposures, and we often don't have them on a granular enough level. So we don't have good ways of studying the impact of air pollution, for example, because we don't monitor air pollution at a big enough scale in enough of counties to be able to, over enough time, to be able to get models for what it actually does do to human populations. And that's changing because getting that data is getting cheaper every day, and the data resources are getting bigger, but we want more. As I alluded to already, if we have data from two different sources, the phenotypes aren't very well aligned. There's little use of ontologies in that space. And there's no organization that I know of that sits down and says, okay, all of you biobank users, can we agree on what a diagnosis of type 2 diabetes might mean? So that if I fit a model in one and I looked at a model for the same disease in a different one, can I compare those models, right? Population structure is often induced. We see a very unusual population structure in a sense in the UK Biobank, where at recruitment it was people, I think, between 40 and 60. All of us in the United States that's trying to recruit 1 million people, tech savvy group, mainly Caucasian, self-selection. NHANES does a different kind of sampling. And all of these really matter when you try to compare things across these. So ontologies give us an organizing principle. So let's see what happens when we do that. So here's an example of an ontology. It's just a formal specification of concepts and relationships between those that try to help us understand what we know. So on the right side here, I just have a little cartoon of cities and structures within those cities, and Paris is the capital of France. So we see these sorts of labels on the edges that tell us about relationships. Great. In medicine, we have a number of ontologies. They're listed in the box on the right that get used. There's OMIM phenotypes, there's OrphanNet, there's SNOMED, NCI thesaurus, genetic rare disease stuff, MESH, medical subject headings, unified medical language, ICD-9 disease codes, ICD-10 disease codes, etc. So we have too many of them. And so whenever we have too many of things, we have an organizing thing. So Mondo, the monarch disease ontology, is the sort of one ring to rule them all, in an analogy to a well-known movie series and book. And we can go into Mondo and we can ask questions like, oh, here, let's look at pancreatitis. And so we see on the left side here, pancreatitis highlighted in blue, and then underneath that, we can see some specific kinds of pancreatitis. And it's underneath, if you look up a little bit, it's underneath something, a heading called pancreas disorder. So great, we have structure. How do I use that structure? All right, so let's map back. So here I have one of the folks that works for me, Rafael Goncalves, sort of helped construct these slides. And so here we can look back and see pancreatitis is a disease of the pancreas. The pancreas has a location. It's an organ in the body. It's an anatomical structure. It's an endocrine disease. It's a digestive system disease. It's an inflammatory disease, etc. And so you'd like to believe that somehow, if I searched in a database for all the inflammatory diseases, I'd be able to get a list, and pancreatitis would be on that list. But sadly, that's not true in, I think, virtually every database around right now. Not picking on OpenGWAS, just using it as a readily available example. So they have basically amalgamated a bunch of GWAS summary statistics for people to use. It's a very large and useful resource. They mapped phenotypes in that. So a GWAS is a study that takes a phenotype and says, does the risk of having that or not having that correlate with a change in a variant in the human genome? And if you pass certain thresholds, then there's a statement that, yes, variation at this location is associated with whether or not you have that phenotype. And so they've mapped the phenotypes to these. Great. And so if I go in there and I search on pancreatitis, I'm in the money. I get a handful of hits over here. But if I remember the list, I could have started with pancreas disorder, and then I could have seen all of the types of pancreas disease that were listed. And sadly, when I do that, I get 0 hits. So we've mapped things to to tags. So we've sort of basically taken that whole system and reduced it down to each phenotype gets a tag. And now I get to search on the tags. But there's no ontology structure behind that. So that's one of the problems that we're working on. How do we solve that problem? How do we make it so that if we know a little bit about the ontology, we can do this searching? All right, switch gears, some omics problems. So what happens when we start doing omics? So genotypes, we just don't have enough of them, certainly not in the public domain. Again, I'll just say, having seen what happens when you have 13 million genotypes and lots of phenotypes, it's really pretty magical, and we would do well in the public domain to try to get up to the tens of millions of people genotyped, and we would learn a lot more. Genes. What we learn is usually on average obtained from bulk processing of cells. We're getting a little bit better. Single cell tells me what's happening in each cell, which is great for cell lines, not so good for tissue. And we'll come to some examples. It's disconnected often from other omic level data, and it tends to focus on mRNA and not on protein. And as most people know, protein is the point of action in some fairly general sense. So we're kind of measuring what we can, not necessarily what we want to. And then really the biggest problem is that we haven't got into the sort of population genomics world yet. We're doing a lot of these studies, especially the CRISPR and other ones on cell lines. And unfortunately, that doesn't tell you as much as you'd like, because you don't have a good sense of what the natural variation in the population is for gene expression. And if you can show that, your perturbations of gene expression change it by a lot inside of a cell line, that's kind of good. But if the population variation exceeds the amount that you can change it by, you don't really have a therapeutic window of any sort, right? And so we're sort of grabbing hold of data that comes out of these very artificial experiments and pretending that it tells us something about what's going to happen in a human in a trial. And same with drugs, right? It's, yes, you knock down this gene in a cell line, but you don't know how much variation there would have been in an outbred population. And that's part of the reason why a number of trials will fail, is because you're able to perturb stuff and it looks like enough the way you're measuring it, but it's just not going to be enough in a population. Opportunities, so the ones that I'll talk about a little bit and show you some examples, because we're working with Jeff Moffat, who's one of the MERFISH folks, are the spatial stuff. So there's been great work done in proteomics, epigenetics, and transcriptomics, and I'll show you some of the transcriptomics and try to get you excited. I think there's a lot of opportunity in organoid systems, especially if we can start to build up, these little tiny, sort of lung epithelial organoids, or sort of a renal type organoids, but using people with different and potentially well-known genetic backgrounds is going to be much more informative than building them up from stem cells from sort of single donors. And I know that's hard, but that's, I think, where we need to go. All right, so let me just run through. So the way that MERFISH works is that we're going to barcode molecules. So here in the middle, we have a cell. It has some little wiggly lines. Those are the mRNA molecules, and I have some barcodes. So I have an RNA. I know the sequence of that, and those barcodes have been designed. And the RNA molecule A needs to have a barcode 110. And so the first thing that I do, and so for each mRNA that I want to identify, I have to make up a library that says, okay, I know the sequence of that, and I'm going to make something that's anti one position, something that's anti a position next to it, and something that's anti a position next to that. I put those they're little cups, actually. I put those in and get them to hybridize to the RNA, and then I introduce to that optically fluorescent markers that will bind differently to the different sort of cups that I put in. So now I've put those cups in there, they're sitting there along each of these RNAs, and now I introduce some probes, and you can see that the probes aligned and show these white or red dots at 4 places, and 2 of the molecules don't have that probe. And then I remove those probes, introduce another probe. Now I can see in the second one that at the top here, we saw a bright mark there. So this probe now, if I'm thinking about its barcode, it's 1, 1. The one next to it is blank. It was bright in the first, so it's a 1, 0. These 2 down here are 0, 0. The 2 that were not lit up in the first are 0 and 1. Image 3, so now we remove those fluorescent markers, introduce new ones, and now we get image 3, and we see that our barcodes are 110, 100, etc. And then we can go back here to the map, because we made it, and we can say, aha, anything that's a 110 was one of these A-type mRNAs. And that's sort of how it works. So the thing about MERFISH is you're making a new library essentially every time you do an analysis. And now you can decode the image. All right, so how does that work? So and then what does it work on it? So it's hundreds to tens of thousands of mRNAs, and it's pretty high throughput. So this is a 2 millimeter tissue slice that you can see lit up with the images, and different mRNAs are being shown, and I'll show you a little bit more in a few seconds here. And then we can actually look in those images. They're very high resolution, and we get subcellular resolution. So we can really point out where they are in the cell. So sort of in analytics, we can figure out if they're in the nucleus or in the cytosol. Lots of fun things. And then you could take that now and just basically, if you just took the cells as they are, counted up the number of mRNAs you saw in each of the cells, you could make a data matrix just like you'd get from single cell, but you have the advantage of being able to map those back to this image on the left and say, oh, here's where they were actually and what part of the image they're in. And that's really what's going to work. And so here we just see a t-SNE map of those as if we didn't have the spatial information, so we can cluster just like we would have in a normal single cell. And so here, we can show that the bulk abundance looks a lot like you'd expect to see from doing a microarray experiment. It recaptures those abundances. That we can show that it if we look at these genes, so now what you're seeing is in this mouse colon, we're just looking at different genes expression. So VIL1 has very strong expression patterns, right? And so this is really sort of a key to understanding that can't be attained from single cell data, because unless you go in and sort of micro dissect out different parts of this, if all you do is take it all, dissociate and sequence. So here, this amount of information is just stunning, at least to me. And we can show that it's different in different parts of the, and now we're moving into now how do we tell disease from normal. All right, so that's the end of that. Those are the things that excite me. As I said, I think that the spatial omics and the similar tools for epigenetics and proteomics in single cell models are moving along as well. And so we're really going to be in a nice place in a few years. I'm almost done. Okay, so what's wrong with the funding model? So here's my take on it, and I'm lucky because I'm relatively unneeding for the research I'm doing now of funding, but outside of our sort of philanthropic donors at Harvard. The PI model that's used in most places just doesn't align very well. You tend to want to fund a PI to do the whole thing, and so they have a stack from beginning to end that they want to cover and have enough money to pay for. But that doesn't help us build tools that are broadly useful. And typically the person who's really good at doing the one experiment or developing the one piece of software isn't very good at sharing. And so we don't have good models. And I think it's true for our Bioconductor, Galaxy, and others. The amount of funding is remarkably low, right? It's on the order of like 3 people in Silicon Valley. That's about what you could hire, or 5 people if you were really, sort of lucky. And so it's just a stunningly small amount of money for the output. That's the problem, right? Is how do we do that? How do we get track record for delivering high quality, widely used tools to get consideration in this? Because it doesn't seem to get that now. Groups like All of Us, for example, funded by the US government, basically restrict non-academic and commercial use. This is a pretty big deterrent. It gets in the way of drug discovery, except for the investigators who are academic who use it to do discoveries for drugs, right? But it keeps it out of the hands of pharma, which is, I think, really unfortunate and not helpful to the US economy. This stuff should be in more people's hands sooner. And, in some sense, just as publication should be free, we really ought to be doing a better job of making sure that data from government sources is just as free, which it just substantially isn't. And there's, not saying it's easy, but, these are the places where I think that there really is a need for some concerted effort, and it doesn't appear that there's a lot of will from on high. And then thanks to the folks that contributed to these slides, mostly from CCB, Ludwig, Raphael, Roger Vargas, Tyrone, Jason Payne, and then for our collaborator, Jeff Moffat and his lab, who provided the slides for that part. And I shall stop there. And hopefully I left a couple of minutes for questions.
Thank you so much, Robert. That was fantastic. So we've got a couple questions. One kind of focus on a comment about reproducibility in research. And a really good question from Rebecca in the chat came and said, what do you think the bottleneck is for making research more reproducible?
So some of the bottleneck is the data is not right. So a research paper, the way that I think of it is, the researcher took some data, they transformed it, and given that they did an okay job of that, right, they then tell a story about what that data should tell me. So they should be required to produce the data that they started with for that story and the code for that story. I'm not that worried right now about how did they get from the raw data to that. I know that there are issues there. That's a different kind of problem. I think this is the problem of, you know, as many people have said, you know, a paper in a scientific journal is an advertisement, right? It says, if you start with this data set and you do these things to it, here's what you can learn. And I should be able to start with that data set and apply those things in minutes, not days or weeks or hours, right? And so that's one of the bottlenecks. We often don't put data that's publicly available into a shared database that people can pull from. That's, again, one of the problems. And, you know, Bioconductor's been trying to do some things along that line. Galaxy tries to do some of the things along that line. We do see efforts by AWS and others to have data in common places that everybody can pull from. And the more we do of that, I think the better off we'll be. We do need to be careful about endorsing one cloud environment over the other. I think cloud is great, but people don't realize, you know, it's Amazon makes more money from the cloud than they do from selling things. Microsoft was on its way to bankruptcy until they got successful in the cloud. And, you know, I'm not sure that Google isn't going to have to do that too now that advertising revenues might be taking a hit, right? So it's pretty easy to transfer all of your cash to one of these companies just to be able to do a bit of computing. So we do have to be careful that we don't buy into one and get locked in.
Right, right. Speaking of kind of like unified attempts at trying to work out publishing, what do you think of Quarto? Have you used it before?
I have not. We're currently scheduling to have some of the folks who used to be called our studio come by and give the people in my group a primer on how to get through Quarto. But I think as we move forward, from what I've heard, I think it's a great tool. And right, we want to put more stuff. You know, Wolfgang Huber and Susan Holmes put their book, all of the bioconductor books we wrote, are basically in really primitive sort of S-weave style of things. And I think the more books, academic books that get published that way, the Oscar book from Bioconductor, these are fantastic resources. They really help you understand what's going on. And even more importantly, then you can just jump in and modify it, right? And start saying, well, what if I did it differently? And I think that gets people into doing real research much faster.
Just kind of at your current position, what do you think the center, that CCB is, its role in this space will be in the future?
So our role at Harvard is really pretty broad. The mission statement is essentially to change the way that we do research at Harvard in the medical school, to change the way that we deliver clinical care by organizing data and algorithms better. So we basically work with different labs and researchers who want to learn how to use a new technology. So it's mostly transferring. So we help them on a project, but the idea is afterwards they've learned something new and they're able to use something, or they want a big data set and they want to have it set up or it doesn't have to be big. They just want to have a data set put in a database where then instead of every person in the lab, pulling files and sorting them in Excel spreadsheets, they can sort of change this to a more query the database. Everybody can query the database. Everybody gets the same answer for the same operation now, and the lab can sort of move forward much faster.
Okay. One more question. I'll put this out there before the next break. Where do you see opportunities for the development of R in the future?
I think going along the lines that we have, I think there's going to be a lot more integration. As I said, like if you look at and I'm totally enamored by the MERFISH stuff, right? These are really big images, and the way that people want to look at them is in the browser. So like whole new technologies are needed to be able to do that. But those technologies are really coming along pretty fast, and R can be the glue in there, right? Things like the sort of shiny or R Studio Connect or whatever it's going to be. Apologies for not getting their name right, but those sorts of technologies really will start to drive, I think, what's happening in science, because they just enable people to pay attention to the details that they need to pay attention to to do their science, right? Right now, you spend all of your time paying attention to details that really are irrelevant to the scientific question. You have to get the data in exactly the right shape, and you have to be careful how you sort it, and what you do with missing values and all of those sorts of things. And that just takes intellectual effort. And so by the end of the day, you're exhausted and you still haven't, done your science question. And so I think the more we can do to make that simpler, the better off we're going to be. And sort of, as I said, to find ways to do it once. So part of our role, I think, in the scientific community is that convening function of here's some things that we can do. Can we find agreement? An example is the NHANES data set. So it's more epidemiology, but pharma uses it. A lot of people use it. It's public, but it sits in a whole bunch of really weird files on an NIH database, and everybody that wants to work with it downloads them individually. And so we're working on a Docker instance that has all of them in it, has all of the functionality, and then anybody that wants to write a paper, if they produce an R script, everybody else can do the same thing.
It's very exciting. Just hearing so many common use cases of how R really is gluing so many things together, and at the same time kind of helping us move from what you said, from doing the science of what we can versus what we want to do. That quote is in my mind. I really appreciate the talk this morning. We have a break coming up for 5 minutes until our next talk at 11. But Robert, I just want to say thank you again for a fantastic presentation.